Recording in progress.
Y ha publicado en revistas de muy alto impacto su investigación, tratando de entender cómo estos RNAs pueden impactar en la herencia epigenética transgeneracional. Germano, thank you very much for accepting the invitation and being here.
I understood everything.
Yes, I know. Despite everything, all the travel problems that you have, and really we appreciate your effort to come to Mexico and to be here today.
Amazing. So where I need to be positioned, because I know that they are video recording. I move a lot. Good. Yeah. So thanks very much. Well, first of all, I need to thank one of the students that selected me among the speaker, you. Thank you. So if the seminar is bad, it's his fault. It's a great responsibility. No, but it's very nice. We had very interesting time yesterday discussing. So sorry, it's gonna be the same seminar, but compressed. So, but it's good to listen again. Maybe not. But it's a great. I think it's a great program. What you're doing here. That you have all those cycle of scientists coming from abroad, and they can interact with the next generation of scientists. So I really congratulate to you. Really. And well, now I'm super excited to talk to you about what we do in the lab. Yeah, we are located in Paris in the Institut Pasteur, which is quite known for infection, but we also had a great history for molecular biology, thanks to Jacob and Monod, that you might know about it. But so my topic of the talk is about epigenetic inheritance. So in these fields of investigation, we are trying to understand whether, in addition from our genomes, the two gametes are transmitting something else to the next generation. And in particularly, we are interested in several types of molecules that we are investigating whether they can also be transmitted from the true gametes to the next generation, and perhaps having some heritable function similar to what the DNA is known for. There are several candidates that we study in the fields. Maybe you heard about those histone modification. Histones are core component where the DNA is organized. DNA methylation is a modification that can intervene directly to the DNA. And there are also other type of things that we are interested in understanding their heritability, like RNAs, proteins, lipids, and more and more. The focus on my lab at the moment is primarily on the role of RNA in this process. And more specifically, we study this molecule called small RNAs. And I will tell you more about them in a few minutes. Why those molecules are interested in this process of inheritance is because in contrast to the DNA that doesn't change in the course of our life, those type of modification can actually change based on what we eat, the type of experience in terms of virus pathogen that we encounter in our life, and also the time that we decide to make babies. They might have an impact on the pool of those molecules that might be heritable and therefore have an impact in the next life or even across multiple generations. And that's why, in the field, we are really trying to pursue the idea that maybe and try to understand whether, those molecules can really transmit what we have lived in our life in terms of environmental information. So those are big questions in the fields. I don't think we are super close to have a clear answer, but I think in the past 10, 20 years, we did quite a lot of work understanding whether this is possible at least, okay? And so, of course, working directly with humans is challenging. And In our lab, we are using a model organism that I think is quite peculiar for understanding this process. And this is a worm. It's called Caenorhabditis elegans. This has been used for many outstanding research, including they discover apoptosis, small RNAs. So in our case, it's a transparent nematode. This is like a gigantic version of it, but it's actually extremely, extremely small. This is in fact, animals that grows on a plate. They eat all the same type of bacteria, and they are really, really tiny. I mean, this is a plate that is maybe big like this. So it's very, very small. Why we are interested in using this model for epigenetic inheritance is because there are two important features for us that render very amenable to this topic. One is that is one of the few animals that is an hermaphrodite but self-fertile. So it can make babies alone without the requirement of any other individuals. And so it's quite prolific because one individual like this can make up to 200-300 babies. And obviously those are all genetically identical. I mean, pretty much similar. And that's why, you know, we reduce a lot the contribution of genetic variation, and we can study whether there are some other things that can be transmitted. So this is very useful. And then the second point is the generation time is extremely short because the life cycle development is super fast. And so you can obtain 2 generations in 3 days. And this, again, you will see in some experiment I showed you, is very important because when you want to study phenomenon that lasts for multiple generations, heritability that maybe start in 1 generation and then in after, you can do experiment in a reasonable time, in 2, 3, 4 weeks instead of months or years. So the molecules that I'm interested in the lab are small RNAs. And there is one peculiar reason I start studying those small RNAs since my graduate work. But since the discovery of small RNAs, there was something that intuitively we thought that there might be connected to some epigenetic inheritance. In fact, those molecules were discovered during the phenomenon of RNA interference. Maybe some of you heard about it. There was a Nobel Prize, in fact, conferred to this phenomenon by Craig Mello and Andy Fire. What they discovered is that if you have a double-stranded RNA intermediate, and you can have of any specific sequence that you want, you can inactivate a gene with the same sequence by exposing any animals or cell lines with the double-stranded RNAs. So for example, I told you the nematodes are transparent, so you can have fluorescent tag version of any protein of interest. And basically you just see these worms that are green under the microscope. And so you can do an experiment where you have a double-stranded RNA with part of the sequence of the GFP. You expose the worm to these double-stranded RNAs, and you will see that 100% of animals on the plate will not express anymore this protein, okay? And so the reason why they don't express this protein, those are research from many years and where they identified the pathway, the molecular events that leads to the inactivation of these genes. One key enzyme that we also have in our cell is called dicer. Those are endoribonucleases. They recognize any double-stranded RNAs that are produced in your cell or even that you are exposed to. And then they cut into small pieces of RNAs, which we call the siRNAs. And those are now effective molecules that are loaded into another group of proteins that are very well conserved in all eukaryotes. They are called Argonaut. These are RNA binding proteins that specifically interact with small RNAs coming from this process. And now the small RNAs is simply a guide that by sequence complementarity can scan all the mRNAs in the cell. And if it finds the complementarity, like in the case of a GFP, and we have a target of the GFP, then you can see how this complex now interact with this RNA and produce an inactivation of this RNA by several methods, including RNA degradation and so on. And so this is the phenomenon of RNA interference that is very well known now. But now, what was very interesting since the beginning of those experiments, it was that when they try to get the baby from these worms that are now silent, and you grow on a plate where you absolutely don't have any presence of double-stranded RNAs, you would expect that now the worm re-express the GFP because there is not this molecule that trigger all these silencing events. And surprisingly, in fact, all these worms are remaining silent, meaning that they do not express the gene, the GFP genes, even if there is no double-stranded RNAs. So there is an inheritance of these silencing events that happen in the parental generation. And this silencing can persist for multiple generations. Again, without doing anything, you don't have double-stranded RNA, you have transmission of silencing. And so this is maybe the first example that we know in animals of transgenerational inheritance of a status of a gene that is off in this case. So I'm going to tell you a little bit why this is happening and describing the molecular process because we study molecular biology, and so we like to understand a bit of details of phenomenology. And so this is the work of many, many different labs in the past years. They discovered that basically in the worms, when you have these double-stranded RNAs, the argonaut recruits another factor that cleaves these RNAs, and now it produces an RNA that contains several traits of this UG nucleotide. So this poly-UG traits is very essential to recruit the most important enzyme in all this silencing process, which is called RNA-dependent RNA polymerase. This is an enzyme known by virologists. They serve for the virus to replicate their genome. You do have in C. elegans, in plants, in fungi. So this RDRP has an affinity for these poly-UG-related RNAs that has been generated by these events. And now the RDRP start to make all single-stranded RNAs that are antisense to the target molecules. And so now these single-strand RNAs are loaded into other argonaut proteins. There are many argonauts in C. elegans. And now some are cytosolic, some are even in the nucleus. And so they can lead to the post-transcriptional, but also transcriptional inactivation of the targeted genes. Now, why after we trigger these events, you still maintain the silencing across generation is because essentially this is a sort of an amplification mechanism that can continue even if you don't have any more these initial steps, okay? So this RNA can continuously produce thousands of small RNAs coming from, few initial molecules of double-stranded RNAs. And this is why today we know that, in fact, these animals that come from the parental generation that were triggered, they still inherit quite abundant of these small RNAs. I forgot to tell you that we call 22G RNAs because of the size, and even of this type of substrate. So these are heritable, and in every generation, you re-amplify and you maintain the silencing that has been established in the parental generation. So now this is what we know about this kind of artificial phenomenon of RNAi. But when I started my lab, I was really interested in understanding whether this pathway can even work to regulate process or traits and heritability in worms. So the most, the tissue that we focus are the germline because this is the place where small RNA can be heritable in the next generation. And in fact, this pathway of small RNAs, what they do in animals, they really work in the germline to protect our genome against genomic parasites. So this is in fact an ancestral RNA-based immune system that is evolved to protect against viruses or even DNA parasite like transposable elements. That I don't know if I remind you, those are elements that can jump into your genome and they can be quite deleterious. So this is the main pathway that control these elements in animal germline. And in animals, there are a specific small RNAs that we call piRNAs because they interact with an agonal protein called PIWI. And so these PIWI interacting RNAs, this is really the central pathway that even in humans controls all these repetitive elements in the animal germline. Now, not surprisingly, if you are born without PIWI or piRNAs, you end up being sterile. And this is thought to be caused by the fact that you don't have anymore this repressive mechanism. All your transposable elements start to be upregulated, transcribed, jumping into the germline, causing the germline to collapse, and you have basically infertility. And this is true in many laboratory models that this experiment has been done. However, when we did this experiment in C. elegans, something very strange happened. Essentially, when you do de novo PIWI knockout, the animals are completely fertile. Nothing happened. This is essentially a plot where each individual dots are progeny from one individual worm. I told you wild type, they laid like 200-300 babies. So you see many worms, they always lay this type of things. This is absolutely similar to a wild type. There is no sterility, meaning zero progeny. However, when you maintain this population of PIWI knockout or the wild type generated by CRISPR knockout, you will see now a completely different dynamics. These populations start to gradually lose their fertility until they arrive 12 generations after you have this PIWI mutation, that you finally have the phenotype that we were expecting, the sterility. I remind you that all these animals here, they have 0 piRNAs. They don't have these small RNAs, okay? But still, they manifest this phenotype very later on. So one hypothesis that in the field was made was that, okay, because of transposon, maybe transposon are causing this phenotype. And so, of course, what we did was to look at transposon with my PhD, Georgia, and you do by RNA sequencing. And the results was very disappointing because we have 60,000 individual retroelements in the C. elegans genome, and a really, really tiny fraction get upregulated. And there is also no correlation between this population and this population. They were very, very the same. So basically, nothing is getting upregulated when you remove those repressors. And this is because of how this piRNA works, that I'm gonna tell you briefly here. So we have in C. elegans more than 15,000 individual genes that transcribe for these very short non-coding RNAs called piRNAs. As I told you, those are all diverse sequences. They act basically to recognize whatever sequence is jumping in the genome. The PIWI can recognize those transposable elements, but now the point is that those molecules are not the one that trigger the silencing. And in fact, this is very similar to what I told you about RNA interference. So they recruit similar proteins like the RNA-dependent RNA polymerase, and they start to amplify the signal by making a lot of these small RNAs, loading in this downstream argonaute protein, promoting post-transcriptional silencing, but even transcriptional silencing by this nuclear argonaute. And so if you remember before that the system can work even without the triggering, we think that the same is happening here, that even if you remove the piRNAs, the transposon are still maintaining silenced by those mechanisms, which also promote canine methylation in the end. And so basically you have all the chromatin that is still there and still repressing those elements. In fact, if you remove one of these downstream effectors, for example, here we generated knockout of the nuclear ago, now you see many more elements that start to be upregulated in the genome. However, when we look at the phenotype, these knockout essentially remain fertile. So there is no sign of transgenerational loss of fertility in this knockout. So those experiments that are kind of negative, but tell us that basically maybe the transposon are not the real reason why there is this progressive sterility that are acquired during several generations. And so what we discovered by looking again in our RNA sequencing is that there is in fact a new silencing processing that is occurring in these animals when you remove a silencer. And so we discovered that there are cluster of genes, is a gene family. Those are the histones, which are essential for fertility. They assemble the genome. And those are now, when you remove the pRNAs, they start to accumulate a massive amount of antisense small RNAs. These are all histone genes here, they are clustered. And you can see that the mRNAs is severely downregulated. And so we start to believe that basically it's this silencing phenomenon that is happening upon we remove the pRNAs that is causing this phenotype. One question that we wanted to address is who is silencing these histone genes when you remove the pRNAs, that is a silencing machinery, which is a little bit counterintuitive. And so what we discovered, and I summarized the results here, is that essentially all these downstream effector here, in absence of this pRNAs or PIWI protein, they just relocalize. And so the same downstream effector are responsible for the silencing of these histone mRNAs. And so we tested this hypothesis by removing this component in the context of PIWI knockout. And you can see that if you start with a PIWI knockout that has very high level of these antisense small RNAs, low level of histone mRNAs, if you further remove this activity genetically or by temporarily depleting this protein, you abolish the production of these histone small RNAs, and you now start to re-express those histone mRNAs. So this is in a context of PIWI knockout, right? So you still don't have pRNAs. And so we wonder whether this can rescue the phenotype. And in fact, when you look at the fertility of this animal that have been treated, you know, this is the control population that are PIWI knockout. You have almost sterility. And then you remove this activity here for 1 generation and you obtain very good level of fertility again, okay? So the same experiment you can do similarly by creating a knockout of PIWI and concomitantly removing this activity. And so here we wanted to prevent the sterility. And in fact, now you can see in this other experiment, again, the PIWI knockout after 20 generations, they are all completely sterile. And here 20 generations pass, we remove this activity. The same time that we remove the pRNAs, they are very stable. They never get sterile. So we believe that we managed to prevent the fertility by understanding the molecular mechanism and the heritability. So what this paper is telling you is essentially that the phenotype that we are observing is not due primarily by the mutation of the pRNA per se, but by the heritability of these molecules that are transmitted across generation. And this is a theme that in C. elegans is observing more and more, how the inheritance of small RNAs can even serve to transmit trait across generation. Now, briefly, I want to tell you what we think is happening when you transfer small RNAs to the next generation. This is really now going a little bit into cell biology. So I need to tell you how those molecular events that I showed you early on, where they are into the germline of C. elegans. So this is the gonad. You can do live imaging in C. elegans very easily. You can tag any protein that you want. This is like histone genes, for example. You can clearly see the DNA. And now you see that basically, if you tag all these argonaute protein, PIWI, or even this RDRP, factor degenerate small RNAs, they all localize close to the nucleus, but they are outside. So those are organelles that we call germ granules, which are attached to the nuclear pore. And these are condensates. You had a lecture a couple of weeks ago about condensation. Those are one of the first condensates that were discovered in worms. These are liquid droplets that are forming by phase separation. So all this RNA process that I told you about are happening in those condensates. Now, what is very interesting is that those granules, they are produced in the germline, and the oocytes are transmitting those phase-separated organelles to the next generation. So you see that this is one cell embryo. Those are the condensates that are detached from the nucleus in this stage. And they immediately polarized on one side of the embryo, and they localize in the cell that is the precursor of all the germ lineage, which is already established at 4-cell stage in C. elegans. So this condensate contain all these small RNAs and processes. And in fact, a nice experiment from these 3 different labs, they managed to show that if you interfere with the granule inheritance in the embryo, these phase-separated granules, you stop heritability, okay? You don't have any more RNA inheritance. And so we start to think that maybe the condensate per se might be a sort of protecting environment for the small RNAs to survive to a massive wave of maternal mRNA degradation that is happening during the beginning of embryogenesis. For example, this is a maternal mRNAs that are inherited from the mother. All those RNAs need to be massively degraded during embryogenesis. And in fact, this is the case. This is more than 100 cell embryos. They don't see anymore in any cells. But you see that all the RNAs persist in these condensates. And in fact, we know that if you remove those condensates now, you don't have any more these RNAs. And so we do think that the granules, that's what they do in heritability. They just protect RNA from biochemical activity that can destabilize those RNAs. And this is very important because the mechanism relies on this RNA-dependent RNA polymerase that needs to de novo synthesize and re-amplify those pool of small RNAs in the next generation. And so if you don't protect these molecules at the beginning, basically you lose the heritability. Now we also work a little bit on understanding how this maternal mRNAs is in fact degraded. So who is degrading these mRNAs? So there are several papers in the field of maternal mRNA decay. Small RNAs are involved. So in this case, we discovered that C. elegans use small RNAs that are made in the mother. And in fact, there is another Argonaute proteins. It's called Cecere1, is loaded with small RNAs in the germline that are antisense to all the active maternal mRNAs. And this is basically, served in the embryo to be heritable and to interact with the maternal mRNAs. And because this protein has a catalytic activity, it can cleave the RNAs and basically facilitate this process of maternal mRNA degradation in all the somatic blastomer. And this process is essential because if you interfere with the inheritance of these small RNAs, the embryo will die. They will not make until the larval stages because you remain all this maternal mRNA accumulating in this somatic blastomer. So on one end, you have granules that can protect. On the other hand, you have small RNAs that can also trigger a massive RNA degradation in the embryogenesis. So this is the work of my PhD student, Pierre. Yeah, I skipped this part. So one other things I want to briefly tell you is about how what the condensate are doing in the germline. So we understand the role of heritability in the inheritance. So this is now start to be very interesting in the field because when you start to look where all this activity that I tell you about, like small RNA synthesis enzyme, Argonaute proteins, factors that are involved only in the heritability of the RNAi, they occupy completely different space in the same droplets. And so those are 3 different condensates. They are nicely separated. They all have different activities. Okay, so this is quite puzzling because the argonauts that are the effector of the small RNA loading, they are separated and they are attached to the nuclear pore. So one hypothesis that in the field has been made is that essentially you don't want that in the germline, all the mRNAs that needs to be transcribed and active, they get trapped in this condensate and produce small RNAs and then they become silent because you want them expressed. And so I think in the field we are really looking for what instead is delivering an RNA that you want to be silent in this condensate that ultimately make the small RNAs. And so this is still something very puzzling. So one thing that we thought at the beginning was that maybe this structure of having all those condensates separated, maybe it's not always the case. And maybe there is a way that those condensates are dynamics. And so this is the work of my postdoc, Eric, where he extensively looked at the germline of C. elegans, two of the condensate I told you about, the small RNA biogenesis and the argonaut protein, always separated, you know, whenever you look. But then he finally found a context in the germline where those two condensates are now together. So there is not anymore this separation, but they are one inside each other's, close to the nuclear pore. And so this is intriguing because it suggests that maybe there is something here that is silencing massively. And when he looked at the expression of piRNAs, it's interesting that the place where you find this condensate together is also the place where you have the piRNA expression. And in fact, if you inactivate temporarily the piRNAs and the PW, these condensates are not together anymore. So these experiments are suggesting us that it's an endogenous massive silencing possibly that is happening here. And so Eric wanted to understand what is happening here in this moment of gonad development and germline. And this is the moment where the worms are making sperms and gametes, male gametes. So you can see here, this is again the hermaphroditic gonad. You have the two gametogenic programs that needs to be activated in the same gonads to make sperm and oocyte. The oogenic is confined in this area always. And the spermatogenic mRNAs, you start to transcribe here in all those nuclei. And then during sperm maturation, this is a rapid process. It lasts 8 hours. You see that spermatocytes start to condense their nuclei to finally have mature sperm. This process of condensation is always accompanied by lack of transcription. And so in fact, you can see that all this bulb of transcriptional activation in this cell, precursor cell, they are completely absent here. So when he looked what happened when you remove piRNAs or even the nuclear ago, which is the effector of the silencing process, he observed essentially that now those condensed nuclei, they retain transcriptional foci, they are not silent, and they basically get stuck in development, and there is a huge delay in sperm maturation. So this is a global phenomenon. I mean, he measured all the spermatogenic transcription is upregulated in those nuclear ago. And so, yeah, it's direct. I will avoid these details because I want to tell you something else. But essentially, the story is about how these piRNAs can, in fact, allows these two condensates to cluster together to trigger a massive silencing of endogenous genes. And this process is very important for sperm development and function. So we think that this work is quite important to establish the fact that, you know, all these defense mechanisms can also be co-opted to have quite important developmental function in animals. And now we know more about how example of piRNAs can also have some other function in addition to transposon repression, okay? So this is his paper. Now he has a lab in Bordeaux. So yes, I want to tell you in this last 10 minutes, a new story that we have in the lab that you see that in the past years, we focus the attention of possible heritability in the germline, what they're doing, condensates and so on. But in the field of epigenetic inheritance in C. elegans, now we are starting to appreciate how even environmental traits can also be heritable. So there are a lot of seminal papers where they show that even behaviors, infection, stress can be transmitted across generation, okay? Now, this poses a very important point in the field of how small RNAs that are potentially generated in the soma, that is the tissue where you are experiencing all this infection or whatever you think of, how these small RNAs are triggering some heritability. And so one possibility is that small RNAs from the soma can directly be delivered to the germline and to the next generation, okay? And so this is something that like we really wanted to explore in the lab. And so we were thinking of how these small RNAs can travel. And so we are starting to study now a pathway that is already known for like few decades that is delivering molecules to the next generation. And this is the yolk pathway. So the yolk is a fundamental nutrient found in all oviparous species that makes eggs. Is synthesized in many different organisms depending on the species. In Drosophila is the fat body. Then you have fish, frogs, they have livers. In C. elegans is made in the intestinal cell. This yolk is composed by one specific protein is called beta-allogenin. It has an affinity for lipids. So it's the major carrier of lipids for the next generation. So in this case of C. elegans, it's made in the intestine, it's secreted in this space, it's called body cavity, and it's really in a couple of minutes is rapidly taken up by receptors that are specific for the beta-allogenin genes, taken up in the oocyte and transmitted to the embryo. This is a strain we made with one of the beta-allogenin protein labeled with GFP. You basically see that you can't even see in the body cavity because the process is so rapid that it's rapidly acquired in the oocyte and embryos. So this is the major source of lipids and nutrition that the embryo has that is in a closed environment. They cannot eat, obviously. And so this is supposed to be essential for their embryonic development. So based on this assumption that there are some very important molecules that are transferred from the soma to the germline, we thought naively that to explore whether this is a route that small RNAs can take to move. So the way that we wanted to do this experiment, obviously, is to just focus on this part, that is, you know, the secreted one before taking up by the oocyte. And ideally, we wanted to sequence whatever material is contained in terms of small RNAs and see what is in there. So my postdoc Metali developed the strategy to basically purify those yolk aggregates. So what we did briefly is to inactivate the receptor. So once you inactivate the receptor, you have all the yolk that is accumulating here in the body cavity. So there is no transfer. And so now if we take these worms, we know that the yolk that we purify from them is only the secreted one before taken up by the oocyte. And maybe we can understand if they carry some RNAs, okay? And this is exactly what we did. We did several steps of purification. And then by FACS sorting, we can specifically purify these yolk aggregates. We sequence small RNAs. And we look at the three major classes of small RNA in C. elegans, like the piRNAs I told you about, these 22G, those are the ones made by the RDRP, and the microRNAs. And we basically observed that there was a very large enrichment of microRNAs in this fraction when you compare with the unsorted one. So somehow there is an affinity of microRNAs that goes into these yolk granules. MicroRNAs are important regulator of gene expression. I think you might heard about them. And so we were interested in understanding whether indeed the yolk can transfer microRNA from the intestine to the embryo. So we did 2 experiments that I'm showing you that I think is quite convincing that this phenomenon is happening. So first of all, to prove that the microRNAs are transferred with the yolk, we generated embryos that by temporarily degrading this protein that is the receptor of the yolk, we generated 2-cell embryo here that are either inherit normal yolk or they do not inherit the yolk. And so we sequence small RNAs in this 2-cell embryo because this is a stage where you still don't have zygotically expressed microRNAs. This is happening later on. So we wanted to really sequence the maternal inherited microRNAs, which by the dogmatic field is coming all from the germline, okay? All the contribution of our maternal microRNAs are coming from the mother germline. Now, when we sequence these microRNAs from these 2 populations, you see that if you don't have the yolk, you do lack some microRNAs. Some of them are quite abundant family, so there is a very significant depletion of microRNAs in these embryos that do not receive the yolk. And our interpretation of this experiment is that indeed, maybe those families of microRNAs are also coming with the yolk to the embryo. So the second experiment I show you is that based on this assumption, we wanted to trying to prove that because the yolk is generated in the intestine, possibly these microRNA are indeed coming from the intestine. And so to do this experiment, we decided to inactivate 2 key enzymes in the microRNA biogenesis, but specifically in the intestinal cell. So this system is called Degron. I don't go into details, but essentially allow you to temporarily inactivate with the drug factors in a specific tissue. So now we inactivate in the mother biogenesis of microRNA in the intestinal cell. We collect embryos, and again, the 2-cell embryo that we sequenced, we see similar type of microRNAs that in fact are depleted in the embryo, which I think is suggesting that indeed we do have a pool of maternal microRNAs that are coming from here through the yolk pathway. Do they regulate genes? So this is the job of microRNAs, really to regulate mRNAs. And so we wonder whether there is any genes that might be regulated when you have a worms that come from a mother that has some depletion of this microRNA in the intestine. So when you first look at the 2-cell embryo, that is this stage here, you see that actually the 2-cell embryo that receive less microRNA, they do receive equal amount of maternal mRNAs. So there are absolutely no changes, despite the fact that you lack the inheritance of these molecules. But when you look at later on in development, in these larvae that develop normally, these are normal microRNAs produced in the intestine, but they lack this microRNA in the 2-cell stage. You see that now there are some misregulated genes. The upregulated ones should be the one that in fact are targeted by this microRNA, and indeed this is the case. And what is very intriguing is that all these changes that we observed in larvae, they are coming from intestine because those are intestinal genes. And this is interesting because it was already observed that most of the yolk, this is a paper from the 83, most of the yolk inherited in the embryo, it's still present in a larvae. This is a kind of old images, but if you have a larvae here, just freshly born as this one, the yolk is persisting in the intestine cell. And so we do think that there is a reservoir of microRNAs that can persist for quite long during development, even post-embryonically. And this is why there might be an impact on those genes later on. So finally, I want to tell you why we think you have this transport of microRNAs. So to tell you this, I need to also tell you that there was very interesting reports that shows that in natural population of worms, yolk inheritance is not equally in all the babies. And so depending on the age of the mother, worms can inherit different amount of yolk. you can see that if you have a mother at the day 1 of embryo production, you have a little bit of yolk inheritance, day 2 more, day 3 even more, okay? And so what in this paper they propose is that this differential yolk inheritance is very important, not much for, you know, like just the embryo, but actually post-embryonically to allow larvae to survive even in stressful environment. For example, you can probe the larvae coming from day 1 mother, day 2 mother, day 3 mother that inherit a different amount of yolk. You can see that this now larvae from day 3 mother, they really grow much better. This is like the size of the worm measured by an instrument that we have in the lab in the condition of a little bit of oxidative stress. This is not strong oxidative, it's just a mild oxidative stress to challenge them a little bit. And then you see that, you know, they develop better, the ones that contain yolk, more yolk. And so the assumption in the field is that yolk and lipids provide this sort of phenotypic diversity in genetically identical individual we are talking about, right? And so we wonder whether in fact it's not the yolk per se that is providing these phenotypic advantages, but actually the presence of the microRNA that we discovered. And so we first wanted to sequence microRNAs in wild-type population, but based on day 3 or day 1 motherhood. And now you see that if you compare the microRNA profile in 2-cell embryo from day 3 versus day 1, there is a large increase of microRNAs. Those are the same type of microRNAs that we found in the experiment before that are coming with the yolk, okay? I forgot to tell you that all the green ones are the one that we found in purified yolks. So then based on this experiment, then we wanted to do a final experiment to prove that indeed these microRNA are the one that you need to survive in a challenging environment. So this is a similar experiment that we did for the microRNA depletion. We depleted the mother's microRNAs or control no depletion. Now we look at in the larvae, these larvae develop normally, we don't induce any degradation. So the degradation was only in the mother. And now we can test their capacity to grow in presence of oxidative stress, control population, we don't inactivate microRNA in the mother intestine. They survive, this is not survival, sorry, this is growing better when they come from day 3 mother because they inherit more microRNA, more yolk. And then when we abolish the production of microRNAs in the intestine, there is a lack of irritability of this microRNA. And now you see that this day 3, well, larvae coming from day 3 mother, they don't make it as better as the control population. So this experiment suggests that the microRNAs and not the yolk per se is really the factors that might contribute to this capacity of larvae to grow in a stressful environment. And I want to just finally finish to the fact that not only maternal age regulate this process, but also stresses. And in fact, you can use several types of stresses in the mother. For example, we used osmotic stress in this case. We can see that this type of stress that the mother experiences, they really affect yolk inheritance. There is a large increase of yolk heritability in this embryo. And now when we sequence the microRNAs in this embryo that come from mothers that are exposed to this type of stresses, we also see increased microRNAs, but now those microRNAs are all microRNAs fundamentally involved in all stress responses pathway. And so we believe that this type of heritability is helping the worms to cope again with stressful environment, but I don't want to bother you with more experiment. But the concept here is that indeed this system of transmission of microRNAs from the soma of the mother to the germline is not a normal flux of information, but it's actually something that can deal with the environmental experience of the parents. And so this can help, in this case, progeny to cope with the future environments that the mothers can perceive in their life, okay? And I wanna just finally finish with the video that this is more what we would like to see after. In fact, this transmission I talked to you about is from the intestine of the mother to the progeny. But actually, in this paper from David Gem's lab, they discovered a few years ago that after the mothers make all the embryos, they don't finish to produce the yolk, but they start to secrete from the vulva. This is the vulva of the worms. You might see at some point some puff that is yolk that is secreted. There is now a big puff. Yeah, it's like a volcanic explosion. And so what is very interesting is that this yolk can be eaten by the larvae. In fact, you can co-cultivate larvae that do not have GFP yolk, and you see that this in the intestine of the little larvae. And so we are now trying to see if this yolk that is constituted of the same yolk that is transmitted to the embryo, they should contain the microRNA. Maybe this might be a further source of transmission that is more horizontal, even to generation and not directly coming from the mothers. And this might be another way to transfer those type of information. And by the way, has been claimed in this paper that this is a primitive form of lactation, in fact. And there is a recent paper in Science that shows that even in other more complex worms animal, they do have kind of yolk lactation from the body. And this is maybe drawing a parallelism of what can happen in other species that do not have yolk, but they have lactation. Maybe similar things can happen in that case. So I finish by acknowledge these are the initial member of my lab that really did all the work I presented at the beginning. They were really fundamental to do all this discovery. The two postdocs, they all have now independent labs. And these are the new people. Neve is the PhD student working on the yolk that we are finalizing. Loan is the technician that do all the transgenesis. And these are all new people that we would like to explore even different way of inheritability through small RNAs and a little bit of chromatin. So that's it. I hope it was not too long. But yeah, if you have any question, I'm happy to answer. Yes. You are the moderator. Oh, okay. Yes.
Thank you for the talk. Very nice. Is in C. elegans also the intestine connected with the nervous system, like in other animals?
Yeah.
So I'm just thinking in the possibility that you have some kind of stress to the nervous system.
Mm-hmm.
If something can be transmitted to the intestine, and then the intestine somehow also produces.
Yeah. This is a very important point. So there is a there are evidences that bacteria infection trigger some behavior. So there is certainly a communication gut-brain. We don't know the degree of this communication, what type of molecules they are transferred. There is a paper showing some bacterial RNA that can even enter into the cells and travel, but we don't know much about the connection, the cell biology connection, but there is certainly an impact, brain, gut.
Okay. So, just the other question very fast. So all the mechanisms that you presented here, all are related to microRNAs, but nothing related to chromatin modifications, chromatin structure. There are examples also in C. elegans?
Yeah, absolutely. Absolutely. Okay. Yeah, absolutely. So, well, the microRNAs, as far as we know, there is no communication with chromatin, with some few exceptions, but the small RNAs I talked to you about in the other part of the talks, those do communicate with chromatin. So the one that do trigger the silencing of spermatogenic, for example, is a nuclear AGO. It also promotes canine methylation. But on top of small RNA-mediated chromatin modification, you can have impact on chromatin independent of small RNAs. And this is also something we are looking at the moment. But there are already evidence that, in fact, many histone marks are inherited in C. elegans, primarily repressive marks, I would say, and this is a general theme in other organisms like Drosophila and mouse, whether there is an impact of germline chromatin from somatic stress. This is exactly what we are doing in the lab, but there are not much evidences for this at the moment. Yes.
Antes de la siguiente pregunta, les recuerdo a quien está en Zoom y en YouTube que pueden escribir sus preguntas en el chat y acá las leemos. Adelante, Marta.
Hi, thank you very much for your talk. I wonder whether in C. elegans, when they made the spermatids, they substitute the histones for the protamines. Yes, they do that.
Not at large extent. This is one of the difference between the nematodes and other species. Very low level of protamine exchange. And in fact, you do have a lot of paternal chromatin inheritance in C. elegans.
Because I wonder whether, because you said that the loss of fertility
Yes.
in the PW mutants was because they silenced the histones, the pRNAs.
Yeah. But those silencing happen in the whole germline. So it do not impact only histone biogenesis in the sperm, but also in the oocyte. You know, it's a global phenomenon. Then on top of this, there is this type of regulation of spermatogenic RNAs. Yeah, there is a compaction, but there is not much protamine exchange, in fact.
Because my question in that sense would be whether do you think that at that moment of massive silencing, when the spermatid is going down, do you think that there could be some genes that are not subjected to this silencing?
I see.
Do you think
we haven't looked at those, but it's interesting. Yeah, it's interesting. It's interesting.
Because if there is a protamine gene, that should be active at that moment, no?
I think the majority of the genes should be silent during the maturation of sperm, as far as I know. I mean, there is really global removal of, you have proteins, but transcription there is a global but we don't have the developmental resolution to capture transcriptomes in all these stages of sperm development. This will address your question whether there are regions that are still active, right? Yes.
Thank
you. Thank you. Yeah.
Yes.
Thank you. I can see from your presentation that the reasons why we call small RNAs epigenetic factors. My question is, if in other animals, is there out of speculation evidence that the small RNAs are actual epigenetic factors?
So this is an important point. Thank you for asking. Well, first of all, we need to understand a little bit what we meant for epigenetic factors. So in my view, is factors that can provide irritability. So the irritability that we see during silencing phenomenon, for example, those are due by the fact that these particular small RNAs can re-amplify across generation. So having a system of amplification is, in my opinion, fundamental for this irritability that goes across generation. Species that do not have this amplification system might have hard time to rely on those mechanisms for irritability, unless the small RNAs are communicating with chromatin, and at that point, you might delegate to chromatin inheritance effects that are initially triggered by the small RNAs. So in species that do not have this amplification system, maybe the only possibility I can see is that the chromatin communication might maintain those effects, assuming that, you know, are transmitted through the germline.
Okay, under this view, humans or mammals, it's unlikely that they use
this is
small
exactly
RNAs
the point. I mean, this RDRP enzyme, for example, is missing in all those mammalian species. But There are also, for example, in C. elegans, we don't have DNA methylation. And maybe, in mammals, some mechanism rely more on DNA methylation than in other species. But I still think that RNA might play a role, well, at least in intergenerational transmission, right? I mean, when you are delivering some molecules to the following generation, maybe they do have an impact in early life that can impact even later life. But then it will stop in one generation. It will not go over.
Yeah, the problem is that there are many papers that mention small RNAs as a general factors.
Yeah.
And I think this is a problem.
This is a problem, conceptual problem, because the experiment they do are not clarifying this issue of heritability. I totally agree with you. Yes. There is a question there. This is she wants to. I feel bad. She was one of the first. Sorry. Yeah. Is off. Sorry. Yes. Good.
Thank you. Yeah, I can hear myself. My question was related to the first question about whether the nervous system was connected to the intestine. I was wondering the same thing, but a little more general. So, because from what you showed, it sounds like the only environmental cues that can be inherited are those that can be sensed by the intestine. So I wonder if you have any notion of any other mechanisms that could connect the intestine, so that they receive other
those are completely irrelevant questions. The fact that we focus on the intestine is because we wanted to find already some mechanism that we could easily manipulate, and the York was great for this. We know the receptor, we know how to purify it, and we found something, right? Obviously, this doesn't exclude that other organs can communicate. Now, I will point out that the intestine is not a negligible organ for the worms because, and I think in general for animals, because it's the site where you interact with all the things that you eat, for example. For the worms, is really sensing the environment. Beside temperature, the gut is the primary organ that trigger all those responses, right? So I think it's still a very important organs that can communicate with many others, including the germline. But it's absolutely relevant. There might be potential mechanism that allows the brain to deliver molecules and to be inherited. We just lack experimental setup to prove those things.
But do you think that would be like through the yolk also, or through
Absolutely
a different?
no.
No,
Well,
okay.
Unless there is a transfer from the brain to the gut, and then the gut can take it. We are trying to do some experiment in this regard. We can inactivate, because not all the components of microRNA are depleted when we remove in the intestine. So there might be contribution from muscle tissue or even the neurons. So this will be a little bit tackling what you are asking, in fact.
Thank you very much.
Okay. Yes.
Thank you for the fantastic talk. Small RNAs are involved in different cellular processes,
Yes.
including development, neurodevelopment, etc. Do you think that this massive silencing event is specific for the sperm development, or is a more general biological process?
Yeah, I mean, in our system, we look extensively whether piRNAs could do anything at all. And that's the only thing we observed. And this is similar to a recent study in mice where so there are different types of piRNAs, in fact. So in mouse, you have packeting piRNAs during the meiosis that those are, they don't have any sequence that target transposon, for example, and nobody knows what they do. They might regulate genes. So what they have as of now is that they do have phenotype that involved sperm formation and function and fertility, male fertility, but they still don't know how. So maybe, yeah, the sperm is something that has been coupled for some reason. Well, this is completely speculative. sperma genes are rapidly evolving genes, for example. So maybe there is some way that you need to cope with the mechanism that target all these invasive elements that are kind of new elements in the genome. And so old elements, they might be protected from all this pRNA silencing. And so maybe, you know, the sperm genes that are rapidly evolving, they might be prone to be silenced, but it's totally speculation. Yes.
Before Marta's question, I will read one from.
Okay. Yes.
It's from Dr. Julian Valdez. He asks, what happened in animals that don't have RDRP, like mammals? Do you think that there is a similar system compensating in the gonad for the function of RDRP?
But this is another system, right?
Yes.
That's the question. Yeah, I mean, as we discussed a little bit this point of having chromatin-based inheritance, that might be one key to overcome the amplification that you need. There is a possibility, there are some small RNAs that they found even in the oocyte of mouse, where they come from pairing of genes that are in opposite orientation, for example. They might have, you know, one gene and the pseudo genes, they form a double-strand RNAs and they produce small RNAs. So again, for me, we discussed yesterday with the student, the fundamental part is the abundance of the molecule that you can produce. So assuming that you have in the genome this alternative production of these endo-siRNAs, how they call, from overlapping genes or double-strand RNAs, this can lead to some heritability effects. More difficult is to understand for how long you can keep those heritable effects and whether you can pass to generation, right? And this is difficult to imagine without an amplification system. But yeah, but I mean, you can imagine, there is a recent paper on Science showing an RNA that persists for several days in the neuron. You can protect those molecules, you know, like in the granules, they are protected until there is a new amplification. I think there is there are a lot of things that we can explore before we finally say, okay, there is no amplification system, RNA is out of the game, right?
I'm just curious.
Yes.
In the case where you mutate or abolish the activity of WAGO or HRD1, where you observe the overexpression of the transposons,
Yes.
those elements, what happened to those worms? Are you able to see a phenotype there?
Yeah, I think the retrotransposition in our system, you can observe in a population, some individuals might have defects, dramatic defects, or you might have some weird phenotype in development, but it's not a global effect that you see in every worm. It's more a random effect that you see, you know, based on that event of retrotransposition. In fact, when we did the experiment to restore fertility, some worms did not restore anything. And I think there might be something related to some transposon in that case that created permanent damage and now is irreversible. So there might be some impact on retrotransposition, but you don't see as a global phenomenon in every worm, but more in a isolated individuals.
WAGO and HRD
Yes.
themselves are not related to the transgenerational effect.
No. Only to maintaining a certain level of fertility by promoting this sperm development and function through the repression of the spermatogenic genes. But in general, they never become sterile in this normal condition of growth.
RNA-dependent polymerase, that one is essential for the transgeneration.
Yes.
It was a very nice talk. Thank you. I was wondering, like, you were talking about these germ granules and like their RNAs were like kind of protected like
Yeah.
inside them, but how are they like specifically target and how they are retained within the germ granules?
Okay, that's a very interesting point. So the small RNAs per se, we don't think they are free. So they are in complex with the proteins. And so if you allow the proteins to segregate with those aggregates, granules, condensate, then automatically, you are sorting specific population of small RNAs, right? So the specificity might be given not by sequence per se of the small RNA, but more by the protein and by the way those proteins can interact with those condensate factors. Done? Okay, good.
Recording stopped.